Hello everyone, welcome back. I hope you all are doing good. So today we are going to talk about difference between acid fast and non-acid fast bacteria. So let's start. So before moving ahead, let's first understand that from where these two terms acid fast and non-acid fast have been taken. Then you should know that there was a scientist named as Paul Ehrlich who developed acid fast staining technique to stain different type of bacteria in 1882. Later on, the protocol of acid-fast staining, as originally developed by Paul Ehrlich, was modified by these two scientists, named as Zeil and Nielsen in the year 1883. That's why the modified version of acid-fast staining protocol, what we use even in today's time, is also called as Zeil-Nielsen staining method. Now you should know, based on the response of different type of bacteria to this staining method, we categorize bacteria into two types. One is acid fast bacteria, another type is non-acid fast type of bacteria. So today we are going to talk about some points of differences between these two types of bacteria. So let's see the first point of difference, color retained. So what kind of color is retained by acid fast and non-acid fast bacteria after acid fast staining? Let's understand this with the help of a pictorial presentation. Suppose here we have taken the bacterial culture of unknown bacteria, and after preparation of smear of these bacteria on a glass slide, when we perform acid fast staining, then firstly we add a primary stain to this bacterial smear, what we call as carbofuscin. After addition of carbofuscin, what kind of results we expect? Like this, that all bacteria present in the sample will take the color of this dye. After this step, we are going for the heating step now, right? So why we do heating here? Heating actually ensures the penetration of primary stain, that is the carbofuscin, inside each and every bacterial cell what is present in the sample that we have taken, right? So after heating step, all bacteria will still be having what? Reddish color of the primary stain. In subsequent step, we go for acid alcohol treatment. Now acid alcohol here actually act as a decolorizer. After addition of acid alcohol, what we observe that some bacteria present in this sample, they have still retained the color of the primary stain, that is carbofuscin, and they are appearing reddish to us. On the other hand, some bacteria present in the sample, they have actually lost their color, or they have undergone, we can say, decolorization after addition of acid and alcohol, right? And now in order to stain those bacteria which have lost their color at this step, we add a counter stain, what we call as secondary stain, and that secondary stain is methylene blue. After addition of methylene blue, the bacteria which actually have lost their color in step number C, they have now taken the color of this dye and they are now appearing as bluish in color. Now this result of acid fast staining actually tells us bacteria are of actually what type. If bacterial sample what we have taken, that is having the bacteria, those bacteria which are actually taking the color of carbofuscin and they are appearing red to us, then we will say that particular bacteria type is of acid fast type. On the other hand, if we are getting results like this, that all bacteria in our sample what we have stained in the laboratory are of bluish color, means they have taken the color of methylene blue, then we will say that those are non-acid fast type of bacteria. And for the bacterial culture in the laboratory, if we are performing this staining and some bacteria have taken red color of the carbofuscin and other bacteria are of bluish color, they have taken the color of methylene blue, then we will say that our culture is having both acid fast and non-acid fast type of bacteria, right? Let's move towards the second point, stain retained. So what kind of stain is retained by acid fast bacteria? Of course, they appeared red to us. It means acid fast bacteria have retained the color of carbofuscin stain, which we added as a primary stain, and non-acid fast bacteria appear to us of bluish color. It means they retain the color of counter stain, that is methylene blue, what we added as a secondary stain, right? Let's see the third point of difference, resistant to decolorization by acid alcohol. So here we can clearly observe that the bacteria which have taken reddish color of carbofuscin, they were actually resistant to acid alcohol treatment. It means acid fast bacteria which appear to us reddish in color, they are actually resistant to decolorization by acid alcohol, and non-acid fast bacteria, they are not resistant to decolorization by acid alcohol because they lost their color, or we can say they undergo decolorization after addition of acid alcohol. Let's see the fourth point of difference, acid fastness. So acid fastness, as its name is indicating, acid fast bacteria show the property of acid fastness, and non-acid fast bacteria actually lack the property of acid fastness. Now acid fastness, this we describe in terms of point number 3, means the bacteria which are resistant to decolorization by acid alcohol, they are acid fast, and which are not resistant to decolorization, they lack acid fastness. Let's see the next point of difference now. Presence of mycolic acid, waxes and complex lipids in cell wall. If we talk about acid fast bacteria, then in their cell wall these substances are present, and in the cell wall of non-acid fast bacteria these substances are absent, right? Now I will try to show you the presence of these substances with the help of some schematic diagrams. So if we talk about non-acid fast bacteria, then cell wall in case of non-acid fast bacteria is of two types, gram positive type of cell wall and gram negative type of cell wall, right? Now we are going to have a look on the presence of different type of layers. So let's start from the gram positive bacteria. This grayish in color layer, this layer is representing you the presence of plasma membrane, which is present just outside the cytoplasm, right? And this bluish layer is representing you the presence of peptidoglycan, right, which I have mentioned here like Pg. And in between peptidoglycan and plasma membrane, this greenish layer is of periplasmic space, which is actually not present in case of all type of gram positive bacteria, and if it is present, even then it will be very narrow. Now we are coming towards gram negative bacteria. Let's observe the presence of different layers in gram negative cell wall. Inside, plasma membrane is present just like gram positive bacteria. Bluish layer is representing us the presence of peptidoglycan layer, and this greenish layer is representing us the presence of periplasmic space, which is wider when we compare it with that of gram positive bacteria cell wall. And the outermost layer is very much unique to the gram negative bacteria, which is made up of lipopolysaccharide. So this is all about the presence of different type of layers, or we can say an overview of different type of layers present in cell wall of gram positive and gram negative bacteria. If you would like to further enrich your knowledge about gram positive and gram negative bacteria and their cell wall, then you can check out the previously uploaded video by me on the same channel. Okay, I hope they will surely be fruitful for you. Now we are coming towards the cell wall of our interest, that is acid fast bacteria cell wall. So here we see, firstly, plasma membrane is present just like gram positive and gram negative bacteria. Outside this layer, this bluish layer is showing us the presence of peptidoglycan layer, right? So now we can see the presence of different type of substances, what I have listed here in acid fast bacteria cell wall, right? So let's observe the presence of these substances one by one. So outside the peptidoglycan layer, actually one polysaccharide layer is present, what we call as arabinogalactan, right? Arabinogalactan is the layer which actually act as a linking material for actually the binding of mycolic acid. Mycolic acid, which is a complex fatty acid present in the cell wall of acid fast bacteria and which is very unique to the acid fast bacterial cell wall, and as mycolic acid is absent in case of gram positive and gram negative bacterial cell wall, this mycolic acid is actually linked to the peptidoglycan via this arabinogalactan, right? And outside this mycolic acid layer, some outer lipids are also present, right? In addition to this, complex lipids which we call as glycolipids, named as phosphatidylinositol manocyte and lipoarabinomannan, right? These are other type of complex lipids which are also usually found in case of acid fast bacterial cell wall. So the presence of these actually make the acid fast bacterial cell wall very much different from that of non-acid fast bacterial cell wall, right? So let's move towards our next point of difference, resistance to disinfectants and dry conditions. Acid fast bacteria are actually more resistant as compared to the non-acid fast bacteria, which are less resistant to disinfectants and dry conditions. Now question comes, why it is so that acid fast bacteria show more resistance? So the answer is this, because acid fast bacterial cell wall is actually of waxy nature, or we can say this is high in lipid content, fatty acids and glycolipids that actually make these bacteria more resistant towards dry conditions as well as water soluble disinfectants. Why? Because it becomes very difficult for water soluble disinfectant to pass through these cell walls. Let's see the next point of difference, response to gram staining. So if we talk about acid fast bacteria are actually harder to be stained by gram staining method. That's why there was need to develop a different type of staining method to stain these bacteria, what we call as acid fast staining. And non-acid fast bacteria, they can be stained by gram staining protocols, right? Now, let's understand a bit in depth that why it is so that acid-fast bacteria are harder to be stained by gram staining protocol. Then you should know whatever kind of dyes we use in case of gram staining, they actually cannot penetrate through the cell wall of acid-fast bacteria. Why? Because they are very much high in lipid content. On the other hand, if we talk about acid-fast staining dye, carbon what we use in case of acid-fast staining protocol, that actually is rich in phenol, right? Because of the presence of phenol, this dye gets solubilizes inside the lipid content and gains its entry inside the cell wall of acid fast bacteria and help the bacteria to undergo staining, right? That is the reason that carbon is capable of staining acid fast bacteria, and dyes used in gram staining, they cannot successfully be used to stain acid fast bacterial cell walls. Now, one more point is there. Some acid fast bacteria are like this, which takes gram staining dyes, but if they take gram staining dyes, the results come like this. Either the reaction will be weakly gram positive or the reaction will be gram variable. Sometimes they show gram positive reaction, sometimes they show gram negative reaction. It means results of gram staining are not considerable when it comes to acid fast bacteria staining by gram staining, right? So let's see the next point of difference. Examples. So if we talk about examples, then you should know Mycobacterium and Nocardia. These are very good and very well known examples of acid fast bacteria. Here I have also listed some of the species of Nocardia and Mycobacterium. Now if we talk about non-acid fast bacteria, of course list is long. Here I have mentioned E. coli and Staphylococcus aureus, which are very well known examples of non-acid fast type of bacteria. One more thing I would like to tell you, that acid fast staining to identify acid fast bacteria is very common for Mycobacterium tuberculosis, as it is routinely used in clinical laboratories for the preliminary diagnosis of this tuberculosis causing organism, what we call as Mycobacterium tuberculosis. So I hope this content is going to help you, and if you found this video helpful, then like this video and don't forget to subscribe to our channel. Thank you so much. Keep watching.